My dear students of 11th class medical, and today we will move ahead with our anatomy of flowering plants. And previously we have discussed the various tissues and tissue systems, and in order to better understand the tissue organization of roots and stem, we need to look or study their internal structure by studying their transverse sections. And today we are going to discuss the internal structure of monocot root by studying it through the transverse section. And we have, or your book has chosen the transverse section of dicot, the sunflower as the dicot root, and the monocot root, as well as we will discuss the transverse section of monocot root as well. Though there are not so large differences between the transverse internal structures of dicot and monocot roots, and a few that we will mention here today in our lecture. So stay keen here. As I was telling you, to understand the tissue organization of roots, stem and leaves, it is convenient to study the transverse sections of the mature zones of these organs. We have to take the transverse sections of roots, stem and leaves to study, to better understand the internal design of their tissues. And firstly we will discuss the Dicotyledons root. The transverse section of sunflower root shows internal organization as follows, and this is the transverse section of the dicot root of sunflower. You can see the outermost layer here is the epidermis with the root hairs, then the cortex, then the endodermis, pericycle, protoxylem, metaxylem, pith and phloem. Now the transverse section of the sunflower root shows the internal organization as follows. The outermost layer is the epidermis. And many of the epidermal cells protrude in the form of unicellular root hairs. You must be knowing the function of the root hairs. And the cortex consists of several layers of thin-walled parenchymatous cells with intercellular spaces. Epidermis is followed by the cortex. You can visualize here firstly the epidermis and then many layers of cortex. Epidermis, here some epidermal cells are protruded into root hairs, and this epidermis is followed by the cortex. These cortical cells are parenchymatous cells with intercellular spaces. The innermost layer of the cortex is called the endodermis. Here is the endodermis, in this pinkish layer here. This is the innermost layer of the cortex. It is called as the endodermis. This endodermis for you. And then it consists of a single layer of barrel-shaped cells without any intercellular spaces. That means they are tightly packed. And the tangential as well as the radial walls of the endodermal cells have deposition of water-impermeable waxy material suberin in the form of Casparian strips. Hope you understand this. I will repeat. The tangential as well as the radial walls of the endodermal cells. That means these endodermal cells have a deposition of the water impermeable. We do not allow the water to pass. That is the waxy material suberin in the form of Casparian citrips. Next to the endodermis lies a few layers of thick-walled parenchyma cells referred to as pericycle, and then initiation of the lateral roots and vascular cambium during the secondary growth takes place in these cells. This is in case of the perennial dicots. Perennial dicots, the dicots which live for many years. And then the pith is small or inconspicuous. The parenchyma cells which lie within or between the xylem and the phloem are called conjunctive tissue, and there are usually 2 to 4 xylem and phloem patches. Later, a cambium ring develops between the xylem and phloem. All the tissues on the inner side of the endodermis, like pericycle, vascular bundles, and pith, constitute the stele. You may have a look after the endodermis. There are many, this pericycle, and then xylem and the phloem, that are the vascular tissues, and inconspicuous pith lies in the center of this dicot root. And now we will discuss the structure of the monocot root. In many respects, dicot and monocot roots reveal similar structures. It has epidermis, cortex, endodermis, pericycle, vascular bundles, and the pith. It looks similar to that in case of the dicots. This is the monocot transverse section of the monocot root. And it has epidermis, cortex, endodermis, pericycle, vascular bundles, and the pith. But compared to the dicot root, which have fewer xylem bundles, there are usually more than 6 polyarch xylem bundles in the monocot roots. Pith is large and well developed. This is the main difference between the dicot and the monocot roots. In dicots, you already know that the pith is inconspicuous, but here in the monocots, there is a large pith in the center which is conspicuous, and with more than 6 polyarch xylem bundles in the monocots, whereas in dicots there are fewer xylem bundles, and there is no secondary growth in the monocot roots, as these monocots usually live for one growing season only. Thanks. Hopefully you must have understood all this, and you may go through your notes and books for further studying these structures of the internal structures of the dicot and the monocot roots. Thanks. Have a nice day.